Hello, everyone. This is going to be Chapter 13, Regulation of Gene Expression. The first part of this chapter is going to focus on mutations, and then the second part of this chapter is going to focus on how gene expression or proteins are going to be regulated. One of the words going to talk a lot about is efficiency in that part, but in this first part, we're just talking about mutations. This is really connected to Chapter 12 with protein synthesis, and you might remember that mutations or any permanent change in the DNA sequence. So this is the nucleotide sequence of your genome. When we see a change in that sequence, this is called a mutation. Now, there are a lot of different types of mutations and causes. We're not going to get into each kind of like cause and effect, but we're going to get into the details a little bit. Now, germline mutations, these are mutations that can be passed from parent to offspring. So you can see here the change in mutation or change in the DNA here. It can be passed on from parent to offspring if it's in the gametes or the sex cells. You can have mutations in your body cells. Those won't go to your offspring, but again, germline mutations, they will change because they will change the offspring's phenotypes because they are in gametes, the sex cells, the sperm or the egg. Now, you can have a lot of different types of causes of these mutations. They can be spontaneous, where, again, those DNA polymerases don't work perfectly when replicating DNA, so sometimes an error gets through. You can also have induced mutation caused by mutagens. These are from things like X-rays, radiation, cigarette smoke, different chemicals. There's a lot of different things that can cause these mutations, but basically you can either have these randomly occur or they're induced and they have a specific cause. Our first type of mutation is going to be a point mutation, and a lot of these words that we're going to go over are what they actually are. So like point mutations, they are when a single point or a single nucleotide changes. So you can see in this example, T is replaced with C. This type that we're looking at is called a substitution mutation, when we have one that's being substituted for another. And again, this is just what it sounds like. One nucleotide is changed to a different nucleotide. Now, we have two different types of missense mutations. Missense mutations occur when the mutation causes the amino acid to change. So you can see here we have no mutation, TTC produces AAG, which produces lysine. Now, our two different types of missense mutations are a conservative, non-conservative mutation. This has nothing to do with politics. This just basically looks at whether the properties are the same as the original amino acid or different. In a conservative mutation, you can see TTC was changed to TCC, which changes to AGG, and that produces arginine. Now, arginine has a very similar, you can see here it's basic, a very similar property as lysine. So it probably will react in the same way, so the protein might not change, but it's, again, going to be determined by that protein structure and function. So a conservative missense mutation is when the properties don't change. However, in a non-conservative, the properties will change. You can see threonine here is a different property. It's polar. It's going to have a different, or I should say, it should affect the folding of the protein a little bit differently than this conservative mutation. So that's the difference between the conservative and non-conservative missense mutations. They are going to change the amino acid that the original DNA had. You can see we have a lot of different properties here within those amino acids. You can see we have like the non-polar amino acids, the basic and the acidic, the polar. So if we go from like threonine to a proline, you're changing the property, which is going to probably change how that protein is going to fold. Nonsense mutation. These occur when there is a stop that's produced. This stop mutation, this stop codon will stop the protein from being produced further. So you can see here the ribosome will read this and then stop because the amino acid was changed from CAG to TAG, which again is going to stop that protein from being produced. So again, this is a type of point mutation that can really cause a dramatic effect to the protein and how it's produced with the shape and function. A silent mutation occurs when the mutation doesn't change the amino acid. You can see here TAT and TAC both produce tyrosine, so the protein won't be affected. So silent mutations, you're not going to see any change in that protein structure or function. So I like showing this image. This is a great way to go over all these different types. So again, no mutation, we produce lysine. A silent mutation, you get the same amino acid. Nonsense, you get that stop. instead of the lysine. In the missense, you're changing the amino acid, but in a conservative, it's the same. Non-conservative. All right, to finish out this section, we're going to talk about frame shift mutations. These type of mutations affect the protein structure a lot more. This occurs when one or more nucleotides are going to be either removed or inserted, and this changes the frame or the codon sequence of the mRNA. So you can see here's an original strand. When we insert this nucleotide here, it's going to shift everything over, and you can see this changes all of the amino acids. This is going to dramatically affect the protein. Since it's not just one amino acid that's changed, you're changing all of them after this frame shift mutation. So this is going to have a much greater effect on the protein structure and function. So the two different types of frame shift mutations, I'm smiling because they are exactly what they sound like. Insertion, you add a nucleotide. Deletion, you remove a nucleotide. And again, this just changes the codon sequence, which changes all the amino acids, which is going to greatly or dramatically affect the protein structure. Now, I want to talk a little bit to end this chapter about what the actual mutations do. So if you have your normal gene with your protein, you can see it's that normal function or that normal structure and function. When you have a mutation, this is either going to change that protein structure or it's going to produce no protein. So what can happen is you can dramatically affect a protein structure, which dramatically affects the function, or it can have very slight effects where it might change it for the better. So it depends on the mutation, it depends on the protein, it depends on the function. There's a lot of different things that it depends on here, but basically what these mutations do is they change the structure and function of proteins. All right, I'm going to quickly go over 13.2, which is about chromosome mutations. These occur when there are larger scale mutations that we find in chromosomes, not just a single or a couple nucleotides. And again, they sound exactly like what they do. Duplication, you can see part of the chromosome is repeated. You can see in a duplication mutation, you get a second B section. Now, it might not seem like this is, I don't want to say bad, but like something that can dramatically affect a person. But having the correct or the right, I don't know if I want to say it that way, but the you don't want to have too much of a gene because that overexpression of a gene can cause problems within a person. Deletion, it sounds like what happens. You can see this is when a fragment of the chromosome is lost. You can see B is lost here in the deletion mutation. An inversion mutation is when the DNA sequence is reversed. You can see the DNA sequence is still there, but it's reversed. It's A, B, C, D, E, F there. Doesn't make sense because it's not the sequence of DNA. Insertion, this is when you insert a part of a chromosome to another portion. You can see how this E was inserted in this chromosome. Again, this can cause problems because chromosomes and DNA need to be in a certain order. And a translocation, this is where you can get a flip-flopping, where you can see here the D was flip-flopped and the E and F were also flip-flopped here. So this is a translocation type of mutation. So these have sometimes more dramatic effects to a person. And I'll go over in class some examples of these, but these are just some types of chromosome mutations you should know. All right, this next chapter is going to be on prokaryotic regulation. And the next two whole sections are really on how your genes are going to be regulated, how that expression is going to be regulated. So it's not that every cell makes every protein all the time. We have to regulate to make our cell production or our protein production efficient. So this first section is more looking at prokaryotic cells, and it's a little bit more simplistic and a little bit more simple with how they regulate their gene expression, but this wasn't discovered until the 1960s. So if you've been noticing, we went from Mendel, we went to the early 1900s with DNA, middle 1900s with the discovery of the structure of DNA. Now we're in the 1960s, we're seeing how the production of proteins is regulated. So we have this thing called the operon model, and this is how prokaryotes or bacteria regulate their protein production. And we have four major parts. We have the regulator, the promoter, the operator, and the genes. I always remember this as PROG, P-R-O-G. I don't know if that will help you, but these four parts are really important when talking about how the genes are regulated in prokaryotes. So let's look at our first site, promoter. It sounds just like what it does. It's the site where RNA polymerase is going to bind to. So you can see this promoter site. We talked about it a little bit already, but basically this is a site where the RNA polymerase can really bind effectively to. Now, we're going to learn about transcription factors. These will help in eukaryotic cells, but in prokaryotic cells, you just have this little section of DNA that says, hey, bind here and start transcription. Genes, this is our next portion here. These are just any genes that are code for some protein. We already know genes code for protein, so this should be, pretty easy to understand. But one thing to understand here is that a section of genes might actually have several genes. So you can see gene A, B, C, D produces proteins A, B, and C, and D. So there might be more than one gene after a promoter. The regulator gene. So we have our repressor and we have our regulator gene. So our regulator gene is going to be found outside the operon. What happens is this creates a DNA binding protein that acts as a repressor. The gene here makes a repressor, which is a protein that's going to stop RNA polymerase from transcribing a gene. So our whole goal here is to activate and deactivate protein synthesis during this time. So this repressor will actually stop RNA polymerase from transcribing some gene. Our operator is going to be the location where the repressor is going to bind to. When the repressor binds to this operator sequence, it's going to stop RNA polymerase from transcribing. So you can see here our operator. It's usually within or next to the promoter because it's going to, again, stop this RNA polymerase from binding, which would then stop transcription. So again, the repressor binds the operator. The repressor is a protein while the operator is a sequence of DNA. So again, let's take a look at our operon model. We have our promoter, we have our operator, we have our genes. Now these genes make mRNA, which make proteins, and these proteins can be used for a number of different things within prokaryotic cells. But again, promoter is where the RNA polymerase binds to, the operator is where the repressor binds to, and this regulator gene is going to make that repressor, and it's found outside of the operon. Now, we have two different types of operon models, and our first type that we're going to talk about is called the lac operon. Lac stands for lactase or lactose, and this is how the prokaryotic cells are going to break down certain sugars. Remember, lactose is a disaccharide, and you're going to have a enzyme called lactase that is going to break down lactose. Now, lactase breaks down lactose into two simple sugars here, galactose and glucose. Our next operon that we're going to talk about is called the trip operon. Trip operons talk about the amino acid tryptophan. So what happens in these prokaryotic cells is we have a bunch of genes that are going to make a bunch of proteins that are needed for the production of tryptophan. Whenever there are low amounts of tryptophan in these cells, what's going to happen is the RNA polymerase will transcribe these genes, making proteins that make tryptophan. So again, the cell needs tryptophan. There's low amounts. The cell is going to transcribe genes that make the enzymes that make tryptophan. Whenever there are high levels of tryptophan, let me go back. When there are high levels of tryptophan in the bacterium, Escherichia. I don't know why it's not going back for me. There we go. When there are high levels of tryptophan, what happens is the tryptophan is going to bind to the repressor. Once that happens, the repressor then will bind to the operator, and it will stop RNA polymerase from making more genes to make more tryptophan. This is showing us that, hey, we already have a good amount of tryptophan. Why make more? It's going to bind here to the operator, which stops transcription, which stops translation, which stops the proteins from producing tryptophan. So we don't need to have more tryptophan in the cell. Now, we have two different types of these operons. This is called an induced operon. This is an operon that's normally off but can be activated when there is a presence of certain substrates. This is the lac operon, where it's usually off, but then it can be activated once we have certain substrates. Our other type is called the repressible operon. This is when the operon is usually on, but when the product is present in high enough amounts, it's going to be turned off, and that's our trip operon. So you have to know the difference between a repressible and a inducible operon. All right, the last section in this chapter is on eukaryotic regulation, and this is where it gets kind of really complicated. If you thought prokaryotic regulation was difficult, this is a little bit more difficult. The great thing is you really only have to understand what each of these is. I'm going to give you examples of each of these, but basically this is how eukaryotes regulate their gene expression. There's 5 different types. You have chromosome structure, transcriptional control, post-transcriptional control, translational control, and post-translational control. And again, I'm going to go over each one of these a little bit in depth. You just have to know kind of basics about each. So chromosome structure. What we can do with our chromosome is we can either tightly pack it with these things called methyl groups. These tightly pack the DNA around those histones. That way, this makes the RNA polymerase unable to bind to a certain gene. So you can see methyl groups were placed around these DNA, so they wrap tightly around the histones. RNA polymerase can't reach it, so it's not going to be able to transcribe it. On the other end, you have acetyl groups. When they are added, they actually loosen the DNA, and you can see here this opens up the DNA for transcription. So this is how the chromosome is structured, is produced. So this is why, again, we're turning off genes and we're activating genes down here for transcription. Now, this is our first type of regulation in eukaryotes. We also have epigenetic controls, and I'm going to show a video about this, but this is basically how your environment kind of changes how DNA is expressed. This is something very new. I actually didn't get to taught this in college. This is a very new field. But basically what you have are these markers on your cells, in your cells, in your chromosomes, that kind of control how transcription occurs. So there's not a lot known about this right now. So we're still finding out a lot about how your environment plays on your gene expression. So the video does a much better job. I'm not going to show it obviously here, but that's the first level of eukaryotic regulation. Next, we have transcriptional control. And this is the most crucial. So if you think about why this is the most crucial, we want to stop things early on so there's not this cascade of other things that happens. It's not going to be efficient. So we stop early on transcription, so translation doesn't occur, and mRNA isn't already produced. So we want to control how much mRNA is produced. We have things called transcriptional factors, and these are proteins that are going to regulate how much gene expression there is or how much proteins are produced by controlling if RNA polymerase can bind to certain genes. You can see here we have these transcriptional factors. They can either activate or deactivate RNA polymerase to produce a mRNA sequence. Again, for every one of these, I show you a different video, but basically you have two different types of these transcriptional factors. And just like the notes say, activators activate and repressors repress. So activators will help transcription, repressors will stop transcription. Very easy. You also have things called enhancers and silencers, but again, they do the same thing. All right, so that was transcriptional control. This is post-transcriptional control, where, again, we've already produced the mRNA. So what's going to happen is we have this long segment of mRNA, and it's called pre-mRNA. What's going to happen is you're going to cut out certain sections of the mRNA. The exons are going to leave the nucleus while the introns are going to remain in the nucleus. You can see here we're taking out the introns. The exons are going to exit the nucleus. So these are it's a mature mRNA strand, but basically we're going to cut out certain sections of the RNA. Now, I know you're thinking, why do we do this? And you can see this is called a spliceosome that does this. This is an enzyme that's going to take out those introns. Why do we do this? Well, what this allows us to do is we can have something called alternative splicing, where we have different numbers or different combinations of exons that produce different proteins. You can see protein A and protein B have different sequences here from this original mRNA strand. What this allows us to do is produce a lot of different proteins from just one gene. So it's not one gene produces one protein. One gene can produce many different proteins. So what that does is it increases our complexity of our proteome, which is our protein genome, but basically allows us to produce a lot more proteins and only have a few genes. When we first were looking at our genome, we thought we had hundreds and thousands and millions of genes. We actually have the same number of genes as a chicken. This is because the complexity increases because we can get different combinations of these proteins. All right, our last couple slides here, post-transcriptional control. The size of the mRNA strand can also affect the speed at which the mRNA leaves the nucleus. So you can have we have a lot of these genes being around 2,000. You can see that bell curve here. Some genes, are up to 5,000 base pairs, but the longer a gene is, the longer it's going to take to get through the nuclear pores of the membrane, which again is going to regulate how much transcription and I should say how much translation occurs. Since the mRNA is already produced, this falls under post-transcriptional control. All right, now translational control. So this can be really complicated. Basically what this does, we have this, it's called RNAi, and there are a lot of different RNAs here, but once the RNA is already produced, you have this certain type of RNA that can actually interfere with RNA and translation. So what happens is these RNA, it's called RNAi, will interfere with how much production of proteins there are. So what happens then is it will tag certain RNA to be degraded. So, if you have an RNA strand that's already been translated, why keep it around? Because it might be translated again. So what your body does is it actually regulates how long the lifespan of this mRNA is. So it's not just the mRNA stays there forever and just keeps on being produced into proteins. What happens is that RNA is then degraded by something called RNAi. All right. So with that translational control, one thing that we have to understand is the mRNA is going to have different lifespans. For prokaryotes, it is very short, and it's because the environments of prokaryotes can change very drastically, and they need to be able to change with that drastic change in the environment. So the lifespans of these mRNA strands are a lot shorter for prokaryotes. For eukaryotes, it's a little bit longer because they can kind of survive those internal conditions. They can survive longer. So sometimes these mRNA strands can survive for weeks on end and without being degraded because we use so much of it. To give you an example, we make 2 million red blood cells every second. So we're producing a lot of that mRNA to produce those red blood cells. You need that mRNA to stay around for a while, so we produce the hemoglobin very efficiently. Our last type of control is translational control. What happens is the mRNA it's called a GTP cap, and what this does, it helps the ribosome to attach to the mRNA. It will also have something called the poly A tail, which is a protection kind of for the most important part of the mRNA. And this, again, is going to kind of determine how much translation occurs because of these two things. After the translational control, or after translational control is post-translational control. So after these proteins are produced, what's going to happen is they can be modified further, and they can actually be degraded down. So again, we already produced a bunch of proteins. What happens is if your cell doesn't need those proteins, the proteins will break down by certain other molecules. And one of those molecules being, there we go, a protease or a proteasome. These are just enzymes that are going to break down proteins. And we do that by tagging them with something called ubiquitin. Ubiquitin is a marker for a protein to be degraded. So whenever you have proteins that you don't need anymore, they're going to degrade by being tagged by ubiquitin, and either a protease or a proteasome is going to come in here and break them down further because, again, the proteins aren't needed, so why keep them around? I think this is going to end this is our end. I love this little meme here, ubiquitin, and you can see the proteasome saying, hey, get in, this because this is where the proteins are broken down.